Welcome to this episode on microfluidics, microdroplets, and microcapsules, science and applications in agriculture. I am excited to have an expert, Professor Gibbs, with me, who will share insights drawn from years of rigorous research and practical field experience. Welcome to our podcast.
Thank you for having me. I'm delighted to be here discussing how microscale engineering and innovative chemical formulations are reshaping modern agriculture. I look forward to exploring the scientific principles behind these technologies, the challenges we face in transitioning from lab to field, and the tangible benefits they can bring to sustainable crop production.
Let's start with the basics. For those new to this subject, can you explain what microfluidics, microdroplets, and microcapsules are and how they differ from traditional agricultural methods? I'm particularly interested in understanding how manipulating fluids at such a small scale can make a practical difference in a field setting.
At its core, microfluidics is the science and technology of manipulating very small amounts of fluids in channels that are typically tens to hundreds of micrometers in size. This allows us to work with volumes ranging from microliters down to picoliters, which is a dramatic reduction compared to conventional methods. Microdroplets are tiny droplets generated within these microfluidic devices, and each droplet can act as an independent microreactor. This capability permits thousands or even millions of parallel experiments in a fraction of the time and resource cost of traditional techniques. Microcapsules, by contrast, are engineered tiny capsules designed to encapsulate active ingredients such as fertilizers, pesticides, or beneficial microbes, and release them in a controlled manner over time. Unlike bulk mixing or spraying, these technologies provide precise control, reducing waste and environmental runoff while delivering active compounds exactly when and where they are needed.
I understand the scientific principles, but I'm curious about the practical advantages. Isn't it a bit of an over-engineered solution for farming? Traditional methods have worked for decades, and some might argue that these high-tech approaches are too fragile or complicated for real-world agricultural applications.
That's a valid concern, but the benefits are substantial and increasingly necessary given today's challenges. For instance, traditional methods like large-scale spraying often lead to inefficient use of chemicals, with significant amounts lost to runoff or degradation before they can be effectively absorbed by plants. Microfluidic systems enable precise dosing, which means that microcapsules can release nutrients or pesticides gradually over time, matching plant requirements and minimizing environmental waste. Additionally, microdroplet-based assays allow for high-throughput screening of various treatments on seeds or plant tissues, accelerating the development of improved crop varieties. Far from being over-engineered, these methods offer a pathway to precision agriculture that can optimize yield, reduce environmental impact, and enhance resilience in the face of climate variability.
Let's delve deeper into microfluidic devices. Can you describe how these devices are constructed and what physical principles govern their operation? I'd like to understand both the engineering challenges and the science behind achieving such precision.
Microfluidic devices are typically fabricated by etching or molding networks of tiny channels into materials like glass, silicon, or polymers such as PDMS. One of the fundamental principles at work in these systems is laminar flow, which is characterized by a very low Reynolds number. This means that viscous forces dominate and the fluid flows in a smooth, predictable manner. This predictability is essential when you want to mix reagents, control reaction times, or form uniform droplets. In many devices, specialized geometries, such as T-junctions or flow-focusing regions, are used to break an incoming fluid stream into discrete, uniformly sized microdroplets. By finely tuning the flow rates and channel dimensions, engineers can control droplet size and frequency with remarkable precision. These systems are often integrated with sensors and feedback loops so that any deviations in flow or droplet formation are automatically corrected, ensuring consistent performance even in complex applications.
That explanation is quite clear, but I remain skeptical about how such precision holds up outside of a controlled lab environment. Many agricultural settings are far from sterile, and conditions in the field can vary widely. How do you ensure that these delicate devices continue to operate reliably when exposed to the rigors of outdoor conditions?
You're absolutely right. The transition from lab to field is one of the major challenges in deploying microfluidic systems in agriculture. In the laboratory, we work under controlled conditions, but on a farm, factors like temperature fluctuations, dust, humidity, and mechanical vibrations come into play. To address these issues, researchers are developing microfluidic devices using more robust materials and protective housings. For example, newer polymers that resist UV degradation and chemical corrosion are being used to construct the channels, and devices are often encapsulated in rugged casings to shield them from dust and physical shocks. Moreover, by integrating on-chip sensors that monitor environmental parameters, the system can adjust operational conditions in real time. These improvements are paving the way for field-deployable microfluidic systems that maintain the necessary precision while withstanding the unpredictability of outdoor environments.
That's promising, though it sounds like there are still significant hurdles to overcome. Now let's shift our focus to microcapsules. How are microcapsules formed within these microfluidic systems, and what are the key advantages they offer compared to traditional bulk formulations?
Microcapsule formation in microfluidic systems typically involves the controlled interaction of two immiscible phases. In one channel, you have an aqueous solution containing the active ingredient. This could be a fertilizer, pesticide, or biostimulant. In another channel, a polymer solution or oil phase is introduced, which will eventually form the capsule shell. At a carefully designed junction, shear forces cause the aqueous phase to break into droplets, and these droplets become encapsulated by the continuous phase. A subsequent step, often involving polymerization or cross-linking, solidifies the shell around each droplet, creating a stable microcapsule. The major advantage of this approach is that the resulting microcapsules are highly uniform in size and composition, which is critical for controlled release application. This precision ensures that active ingredients are protected from premature degradation and are released at a controlled rate. As a result, plants receive a steady supply of nutrients or pesticides over an extended period, reducing the need for repeated applications and minimizing environmental impact.
I can see the theoretical benefits, but can you provide an example of how such microcapsules have been used successfully in the field? I'm interested in hearing about real-world data that backs up these claims.
Certainly. Consider a case study from cereal crop production where nitrogen-based fertilizers are a key input. Traditional nitrogen fertilizers often suffer from losses due to volatilization and leaching, which not only reduces efficiency but also contributes to environmental pollution. Researchers developed microcapsules encapsulating a nitrogen-rich fertilizer engineered to degrade gradually in soil conditions. Field trials demonstrated that crops treated with these controlled solution microcapsules exhibited significantly higher yields compared to those treated with conventional fertilizers. The steady release of nitrogen matched the nutrient uptake pattern of the crops, resulting in more efficient use of the fertilizer and reduced losses through runoff. Additionally, the gradual release helped to maintain soil health by avoiding the peaks and troughs of nutrient concentration that can stress plant systems. This example underscores the potential of microcapsule technology to transform agricultural practices through improved resource efficiency and environmental stewardship.
That example is compelling, yet I'm still curious about how these technologies integrate with digital tools. In today's age, data is king. How do data analytics and artificial intelligence play a role in optimizing microfluidic applications in agriculture?
Data analytics and AI are absolutely central to the advancement of microscale agricultural technologies. In high-throughput screening systems, such as those based on microdroplets, you can generate vast amounts of data from thousands or even millions of parallel experiments. For instance, when screening for optimal seed germination conditions or testing various nutrient formulations, automated imaging systems capture detailed information about each droplet's response. Machine learning algorithms then analyze parameters like fluorescence intensity, growth kinetics, and morphological changes to determine which conditions yield the best results. This real-time analysis enables researchers to rapidly optimize experimental conditions and make data-driven decisions. Moreover, when microfluidic sensors are deployed in the field to monitor parameters such as soil moisture, nutrient levels, and pathogen presence, the data is transmitted wirelessly to central systems. Here, AI algorithms process the data to provide actionable insights, such as adjusting irrigation schedules or fertilizer application rates, thus enabling a form of smart farming that adapts dynamically to changing conditions.
I'm intrigued by the promise of these digital integrations, but I remain cautious about scalability. For large-scale agricultural operations, data management can become overwhelming. How do you address issues related to data volume and the need for real-time processing in such systems?
Scalability and real-time data processing are indeed critical challenges when deploying these technologies on a large scale. The key is to leverage cloud computing and edge processing. In many modern systems, data collected by microfluidic sensors is first processed locally or at the edge using embedded processors. This initial processing filters and aggregates the data, reducing the load before it is transmitted to a central cloud server. Once in the cloud, more sophisticated AI algorithms analyze the data in real time, integrating inputs from multiple sensors across a farm. Standardized data formats and interoperability between different devices are also crucial for ensuring that data from disparate sources can be easily combined and analyzed. With these approaches, even very large datasets can be managed efficiently, enabling rapid decision-making that is essential for precision agriculture.
That all sounds very futuristic. However, many in the agricultural community are skeptical about the cost effectiveness of such high-tech solutions. Given that farms often operate on thin margins, how do you justify the initial investment required for microfluidic and microcapsule systems?
It's a legitimate concern that the initial investment can be substantial. However, when you consider the long-term benefits, the return on investment becomes quite attractive. For example, microfluidic systems operate with extremely small volumes of reagents, which drastically reduces the cost per assay or application compared to traditional methods. Additionally, the increased efficiency in resource use, such as the more effective delivery of fertilizers or pesticides, can lead to significant cost savings over time by reducing chemical usage and labor expenses. Field trials have consistently shown that crops treated with controlled deletion microcapsules require fewer applications, which not only lowers input costs but also reduces the environmental impact, potentially avoiding fines or compliance costs related to pollution. Furthermore, as manufacturing processes improve and economies of scale are realized, the unit cost of these advanced devices is expected to drop considerably. In many cases, government grants and subsidies for sustainable agriculture can help offset initial costs, making these technologies accessible even for smaller operations.
I appreciate that explanation, but let's talk about the scientific rigor behind these technologies. Some critics argue that the miniaturized conditions in microfluidic devices might not accurately reflect the complexities of field environments. How do you ensure that the experimental results obtained in microdroplets or microcapsules are truly representative of what happens in a real agricultural setting?
That's an important point. In any screening system, it's crucial to recognize that the controlled conditions in a microfluidic device are a simplification of the real world. However, the purpose of these systems is to serve as an initial high-throughput screening tool. They allow us to rapidly identify promising candidates, whether they be crop varieties, nutrient formulations, or biocontrol agents, that can then be validated under more realistic conditions. To bridge the gap between laboratory and field, researchers often design microfluidic experiments to mimic key aspects of the natural environment. For instance, when screening for drought tolerance, microdroplet assays can be configured to simulate varying water availability and stress conditions. Once promising candidates are identified, they are subjected to rigorous field trials and larger-scale experiments. This tiered approach ensures that while the microfluidic system provides rapid, reproducible data, the final validation occurs under conditions that closely resemble the complexities of real-world agriculture.
Let's pivot a bit to discuss some of the challenges. What are the main technical hurdles you encounter when scaling these microscale technologies from the lab to the field, and how are you working to overcome them?
There are several challenges in scaling microscale technologies for field use. First, there's the issue of device robustness. In the lab, environmental conditions are controlled, But in the field, devices must contend with temperature extremes, dust, and moisture. To address this, we are developing more durable materials and protective enclosures that can shield the sensitive microfluidic components. Second, there is the challenge of mass production. Techniques such as soft lithography are excellent for prototyping, but are not ideal for large-scale manufacturing. We're exploring alternative methods like injection molding and roll-to-roll processing to produce devices in higher volumes at a lower cost. Third, the integration of these devices with digital networks presents its own hurdles. Ensuring reliable wireless communication, data security, and power management in remote settings requires a multidisciplinary approach. Researchers are partnering with experts in IoT and embedded systems to create comprehensive solutions that integrate seamlessly with existing farm management systems. Through these collaborative efforts, we're gradually overcoming the barriers to widespread field deployment.
Some critics say that while these technologies are impressive in theory, they might simply not be practical for everyday farming. What's your response to those who question the real-world applicability of microfluidics and microcapsules in agriculture?
I understand that skepticism, especially when a technology seems to come from a high-tech lab rather than a dusty farm field. However, there is a growing body of field research that demonstrates the practical benefits of these technologies. For instance, microcapsule-based fertilizers have been trialed in various crop systems with remarkable results in yield improvement and resource efficiency. Similarly, portable microfluidic diagnostic devices are already being used in pilot programs to rapidly detect plant diseases and nutrient deficiencies directly on the farm. The key is that these technologies are not meant to replace traditional practices overnight, but to augment and optimize them. By providing precise, data-driven insights and reducing waste, they offer tangible benefits that can make farming more sustainable and cost-effective. Moreover, as these systems are refined and scaled up, their ease of use and robustness will continue to improve, making them accessible to a broader range of agricultural operations.
Let's explore the interplay between microdroplet technology and plant breeding. How do microdroplets facilitate high throughput screening for traits such as drought tolerance or disease resistance? And what impact does that have on the pace of breeding new crop varieties?
Microdroplet technology is a transformative tool for high-throughput screening in plant breeding. Traditionally, assessing traits like drought tolerance or disease resistance involves setting up numerous experiments in multi-well plates or field plots, which can be time-consuming and resource-intensive. With microdroplets, each droplet functions as a miniaturized reactor, allowing you to test thousands or even millions of individual seeds or plant cells simultaneously. For example, in a screening experiment for drought tolerance, individual seeds can be encapsulated in droplets containing precisely calibrated water levels and stress hormones. Automated imaging systems monitor germination and early growth, and machine learning algorithms analyze the data to identify which genetic variants perform best under stress conditions. This high-throughput, highly controlled approach drastically reduces the time required to identify promising candidates. The rapid data generation accelerates the breeding process, allowing researchers to move promising lines into field trials much faster than traditional methods would permit.
I'm also curious about the integration of these technologies with environmental monitoring. Can you explain how microfluidic sensors are being used to track soil and plant health in real time, and how this information can feed back into decision-making for crop management?
Absolutely. Microfluidic sensors are being designed to continuously monitor critical parameters, such as soil moisture, pH, nutrient levels, and even the presence of pathogens. These sensors are compact and highly sensitive, and when deployed in the field, they provide real-time data about the microenvironment surrounding the crops. For instance, a network of sensors can track fluctuations in soil nitrogen levels throughout the growing season. This data is transmitted wirelessly to a central management system that uses AI algorithms to analyze the trends and predict when and where nutrient levels might fall below optimal thresholds. In response, the system can automatically adjust irrigation or fertilizer applications to ensure that crops receive the exact amounts they need. Such a feedback loop not only maximizes resource efficiency, but also helps prevent stress conditions that can lead to reduced yields or crop failure. The ability to monitor and respond in real time is a cornerstone of precision agriculture, and microfluidic sensors are a key technology enabling this shift.
Your explanation about real time monitoring is compelling, but I have to ask, given the inherent variability in agricultural fields, how do you account for the heterogeneity in soil and microclimatic conditions when deploying these sensors? Doesn't this variability make it difficult to derive accurate, actionable data?
That's an excellent and complex question. Agricultural fields are indeed heterogeneous with variations in soil composition, moisture, and microclimate even over short distances. To address this, sensor networks are designed to be densely distributed across the field to capture this spatial variability. Each sensor records data from its immediate surroundings, and when combined, the data provides a comprehensive map of the field's conditions. Advanced statistical models and machine learning algorithms are then used to interpolate and analyze the data, identifying patterns and anomalies that might indicate localized issues. This approach not only compensates for the inherent variability, but also allows for site-specific management practices. For example, if a particular section of a field shows lower moisture levels, targeted irrigation can be applied precisely where needed, rather than watering the entire field uniformly. This level of precision is critical for optimizing resource use and enhancing crop performance in diverse agricultural environments.
Let's discuss another angle, the environmental impacts. Many environmentalists are concerned about the overuse of chemicals in agriculture. How do microcapsule-based systems contribute to more sustainable practices, and what evidence is there that they can reduce chemical runoff and improve soil health?
Microcapsule-based systems contribute significantly to sustainable agriculture by enabling controlled and targeted release of agrochemicals. Traditional methods often involve broadcasting chemicals over large areas, which not only leads to inefficient uptake by plants, but also causes chemical runoff that can contaminate waterways and degrade soil quality. Microcapsules, on the other hand, encapsulate the active ingredient within a protective shell and release it slowly over time, matching the plant's nutrient uptake schedule. This controlled release minimizes spikes in chemical concentration, reducing the risk of leaching into the soil and runoff into water bodies. Field studies have demonstrated that crops treated with microencapsulated fertilizers and pesticides require fewer applications while maintaining or even improving yield. For example, in trials with microencapsulated nitrogen fertilizers, researchers observed a significant reduction in nitrogen loss and improved nitrogen use efficiency, resulting in both economic and environmental benefits. By ensuring that plants receive a steady, optimal dose, microcapsule-based systems support better soil health and reduce the overall chemical footprint of agricultural operations.
I have to wonder about the reliability of these systems in real-world conditions. Have there been instances where controlled release mechanisms failed or did not perform as expected under variable environmental conditions? How do researchers mitigate such risks?
It's true that no technology is without its challenges, and controlled sealing systems can sometimes face issues such as premature release or inconsistent degradation rates under extreme conditions. However, extensive research is being conducted to refine the formulations and materials used for microcapsule shells. Scientists are experimenting with hybrid materials that combine synthetic polymers with natural biopolymers, which can be tuned to degrade in response to specific environmental triggers like soil pH, temperature, or moisture. Rigorous field testing under diverse conditions is also a critical part of the development process. These tests help identify potential failure modes and allow researchers to adjust the capsule formulation accordingly. In addition, many modern systems include built-in sensors that monitor the release kinetics in real time, enabling adjustments to be made on the fly. While there are always risks with any new technology, the iterative process of testing and refinement is steadily increasing the reliability and robustness of controlled release systems in agriculture.
Now, turning our attention to the integration of these technologies with genetic and breeding programs, how do microdroplet systems aid in screening and selecting for traits like drought resistance or disease tolerance? I'm particularly interested in the quantitative aspects and how this data feeds back into breeding decisions.
Microdroplet systems enable high-throughput screening by creating thousands of uniform microenvironments where individual seeds or plant cells can be tested under controlled conditions. For example, to screen for drought resistance, individual seeds can be encapsulated in droplets with precisely controlled water availability and various stress factors. High-resolution imaging and automated analysis then capture key growth parameters such as germination rate, root length, and overall vigor. Machine learning algorithms analyze these quantitative metrics to identify which genotypes perform best under stress. The statistical power of testing thousands of individual samples in parallel provides a robust data set that can reveal even subtle differences in performance. This data is then integrated with genomic information, allowing breeders to correlate specific genetic markers with drought tolerance. As a result, breeders can select the most promising candidates for further development, accelerating the creation of new, resilient crop varieties.
I'd like to push back on one point. Some argue that microdroplet screening is too reductionist, failing to capture the complex interactions that occur in a full field environment. How do you reconcile the controlled conditions in these devices with the unpredictability of the natural world?
That's a common critique, and it's important to recognize the limitations of any in vitro system. Microdroplet screening is not intended to completely replicate the complexity of a field environment. Rather, it serves as an initial, rapid filtering tool to identify promising candidates. The controlled conditions of a microdroplet system allow for the isolation and analysis of specific variables, thereby reducing noise and improving reproducibility. Once potential candidates are identified through high-throughput screening, they undergo subsequent rounds of testing in more complex environments, first in greenhouse settings and then in field trials. This tiered approach ensures that while the microdroplet system may be reductionist, it effectively narrows the focus to the most viable options, which are then rigorously validated under real-world conditions.
Let's talk about cost effectiveness from a scientific perspective. Given the high-tech nature of these systems, what kind of cost savings or yield improvements have been observed, and how do these translate into practical benefits for farmers?
Cost effectiveness is a major driver for the adoption of these technologies. Although the initial setup and research and development costs can be high, the long-term savings are significant. For instance, microfluidic systems operate with extremely small volumes, meaning that the amount of expensive reagents or chemicals required is minimal. In controlled lesion applications, the precise dosing reduces the overall quantity of fertilizers or pesticides needed. Field trials have shown that crops treated with microencapsulated formulations not only require fewer applications, but also demonstrate improved nutrient uptake and higher yields. One study on microencapsulated nitrogen fertilizers in cereal crops reported yield improvements of up to 15%, along with a significant reduction in nitrogen loss. These improvements directly translate to lower input costs, reduced environmental impact, and ultimately a higher profit margin for farmers. The integration of these technologies with precision agriculture tools further amplifies these benefits by enabling real-time adjustments that optimize resource usage across large areas.
Your points about yield and cost improvements are compelling, yet I wonder about the learning curve for farmers. Many traditional growers might be hesitant to adopt such complex technology. How do you envision the transition from conventional practices to these advanced systems? And what measures can be taken to ensure widespread adoption?
Adoption is indeed a critical aspect of the transition to high-tech agricultural systems. One strategy to bridge the gap is through the development of user-friendly plug-and-play devices that require minimal technical expertise. Training programs, demonstration projects, and partnerships between technology providers and local agricultural extension services can help demystify these technologies for farmers. It's also important to present clear, quantifiable benefits, such as increased yield, reduced chemical use, and improved soil health, in terms that resonate with the farming community. Pilot programs that showcase successful implementation in similar agricultural settings can build trust and demonstrate feasibility. Over time, as more farmers experience the benefits firsthand, the technology is likely to be adopted more widely. Additionally, government incentives and subsidies for sustainable farming practices can accelerate this transition, making it economically attractive for growers to invest in advanced systems.
I'm curious about the interdisciplinary nature of these developments. How do you see the collaboration between engineers, chemists, biologists, and data scientists shaping the future of these technologies in agriculture? It seems like a daunting challenge to integrate all these fields effectively.
Interdisciplinary collaboration is absolutely essential to the success of these technologies. In my experience, breakthroughs often occur at the intersection of multiple disciplines. Engineers design and optimize the microfluidic devices. Chemists develop the materials and formulations for microcapsules. Biologists provide insights into plant physiology and stress responses. And data scientists build the algorithms to process and interpret the data. Each discipline brings a unique perspective and expertise that enriches the overall development process. Collaborative research centers and interdisciplinary projects are increasingly common, where experts from different fields work side by side to solve complex problems. For example, a project might involve engineers and chemists working together to create a robust microcapsule, while biologists and agronomists test its efficacy under simulated field conditions, and data scientists develop predictive models to guide its use. This integrated approach not only speeds up innovation, but also ensures that the solutions developed are practical, scalable, and tailored to real-world challenges.
Let's shift gears to discuss environmental sustainability. How do these microscale technologies contribute to a reduction in the environmental footprint of agriculture? And can you share any quantitative data or case studies that support these claims?
Microscale technologies offer several environmental benefits by optimizing resource use and reducing waste. For instance, traditional fertilizer applications often lead to significant nutrient runoff, which can cause water pollution and eutrophication. In contrast, controlled deletion microcapsules deliver nutrients gradually and precisely, matching the plant's uptake pattern and significantly reducing losses. In one case study involving microencapsulated nitrogen fertilizers in cereal crops, researchers observed not only a 15% yield improvement, but also a marked reduction in nitrogen runoff by as much as 30% compared to conventional applications. Similarly, the precise dosing enabled by microfluidic systems minimizes the overapplication of pesticides, thereby protecting beneficial insects and reducing chemical residues in the environment. The cumulative effect of these improvements is a more sustainable agricultural system that maintains productivity while mitigating adverse environmental impacts. The quantitative data from these studies supports the notion that embracing microscale technologies can lead to both economic and ecological benefits.
Some critics argue that the environmental benefits are overstated and that the energy and resource inputs required to produce these advanced devices might offset the gains in efficiency. How do you respond to those concerns, especially from a scientific standpoint?
It's important to consider the full life cycle analysis of any technology. While the production of microfluidic devices and microcapsules does require energy and materials, these inputs are relatively small compared to the overall scale of agricultural operations. Moreover, as manufacturing techniques improve and economies of scale are achieved, the energy and resource costs per unit will continue to decrease. When you compare these costs to the savings achieved through reduced chemical use, improved yield, and lower environmental remediation expenses, the net benefit is significant. Many life cycle assessments have shown that the reduced application rates and improved efficiency of microscale systems result in a lower overall environmental footprint. Additionally, ongoing research in green manufacturing processes aims to further minimize the energy requirements associated with producing these devices. Thus, while it's important to remain vigilant about the environmental costs of new technologies, the current evidence suggests that the benefits of microfluidics and microcapsule systems in agriculture outweigh the inputs required for their production.
I'd like to challenge you on another scientific front. In many of these applications, precision is touted as a major advantage. However, natural systems are inherently variable. How do you ensure that the precision achieved in microfluidic systems translates into reliable performance in the highly variable conditions of an agricultural field?
That's a critical question. In the laboratory, precision is easier to achieve because conditions are tightly controlled, but the field is much more unpredictable. To bridge this gap, our approach is twofold. First, we design microfluidic experiments to capture the range of variability found in natural systems. For instance, when testing drought tolerance using microdroplets, we deliberately introduce gradients of water availability and other stress factors that mimic field conditions. Second, once promising candidates or formulations are identified, they undergo extensive validation in greenhouse trials and then in actual field tests. This multi-tiered testing process ensures that the precision observed in the micro-scale experiments is robust enough to hold up under real-world variability. Moreover, the integration of real-time monitoring systems in the field helps adjust for environmental fluctuations, ensuring that the benefits of precision dosing and controlled release are maintained. This iterative process, moving from controlled screening to progressively more complex environments, helps us refine the technology so that it reliably performs even in the face of natural variability.
Let's consider the future of these technologies. What major breakthroughs do you foresee in the next decade, and how might they further revolutionize agriculture? I'm particularly interested in any emerging research that could push these technologies even further.
The next decade promises to be extremely exciting for microscale agricultural technologies. One major breakthrough we expect is the integration of microfluidic systems with advanced sensor networks and IoT devices. Imagine a farm where microfluidic sensors continuously monitor soil and plant health, transmitting real-time data to an AI-driven management system that optimizes everything from irrigation to fertilizer application. This convergence of physical and digital technologies will create truly smart farms that can adapt dynamically to changing conditions. On the microcapsule front, research is moving toward multifunctional capsules that not only deliver nutrients or pesticides, but also incorporate sensors that report on their release kinetics and local environmental conditions. Advances in nanotechnology and materials science will enable these capsules to respond to multiple stimuli, such as changes in pH, temperature, or enzymatic activity, ensuring that active ingredients are released exactly when needed. Additionally, there is promising research on integrating genomic data with high-throughput microdroplet screening, which will accelerate precision breeding. By correlating genetic markers with phenotypic performance under controlled microfluidic conditions, we can rapidly develop crop varieties that are better adapted to specific environmental stresses. These breakthroughs, coupled with continued improvements in cost-effectiveness and robustness, will further revolutionize agriculture by making it more sustainable, efficient, and responsive to global challenges.
Some of these ideas sound very futuristic. However, the skepticism remains among many practitioners who have relied on conventional methods for generations. How do you propose we bridge the gap between cutting-edge research and everyday farming practices, especially in regions that may be less technologically advanced?
Bridging this gap requires a multi-pronged approach. First, it's essential to demonstrate the clear, quantifiable benefits of these technologies through well-documented pilot projects and field trials. When farmers see tangible improvements in yield, cost savings, and environmental performance, they're more likely to embrace new methods. Second, technology developers must focus on creating user-friendly, robust systems that can be easily integrated into existing farming operations. This means designing plug-and-play devices with intuitive interfaces and minimal maintenance requirements. Third, educational outreach is critical. Workshops, training programs, and partnerships with local agricultural extension services can help demystify these technologies and show how they can complement traditional practices rather than replace them. Finally, policy support from governments and international organizations can incentivize the adoption of sustainable technologies through subsidies, grants, and research funding. By addressing both the technical and social aspects of technology transfer, we can ensure that advanced microscale systems are accessible and beneficial to farmers around the world, regardless of their level of technological advancement.
I'd like to challenge your view on interdisciplinary collaboration. You mentioned that engineers, chemists, biologists, and data scientists all need to work together for success. In your experience, what are the main obstacles to such collaboration, and how can they be overcome in practice?
Interdisciplinary collaboration, while essential, is not without its challenges. One major obstacle is the difference in terminology and research methodologies across disciplines. For instance, an engineer might approach a problem from a design and optimization perspective, while a biologist might be more focused on variability and natural systems. These differing priorities can sometimes lead to misunderstandings or conflicting objectives. To overcome this, it's important to establish a common language and clear communication channels early on in a project. Joint workshops, regular interdisciplinary meetings, and the involvement of project managers who understand multiple fields can help bridge these gaps. Funding agencies are also increasingly recognizing the value of interdisciplinary projects and are designing grant programs that encourage collaboration across fields. By fostering an environment of mutual respect and shared goals, these obstacles can be mitigated, leading to more innovative and robust solutions that draw on the strengths of each discipline.
Let's dive into a specific technical question. In many microfluidic devices, droplet uniformity is crucial. What are the most effective methods to ensure uniform droplet formation, and how do you account for potential variability in the field?
Ensuring uniform droplet formation is a central challenge in microfluidics. The most effective methods involve precise control of the flow rates of the fluids, the design of the microchannel geometry, and the selection of appropriate fluids with well-characterized properties. Devices often use flow focusing or T-junction designs, where the shear forces at the junction break the incoming fluid into droplets. By calibrating the flow rates using high-precision pumps or pressure controllers, we can achieve a very narrow distribution of droplet sizes. Additionally, the use of surfactants in the continuous phase can help stabilize the droplets, reducing coalescence and ensuring uniformity. In the lab, we continuously monitor droplet size using high-speed imaging, and feedback systems adjust flow parameters in real time. In field applications, while conditions may be less controlled, the design principles remain the same. Robust microfluidic systems incorporate sensors and automated adjustments to maintain droplet uniformity even when minor environmental variations occur. This combination of precise engineering and real-time control ensures that the droplets remain as uniform as possible, which is critical for reproducibility in high-throughput assays.
I'm particularly interested in the application of these systems for pathogen detection in crops. How do microfluidic devices enhance the detection of plant pathogens compared to conventional methods? And can you provide a concrete example?
Microfluidic devices offer several advantages for pathogen detection. Traditional methods, such as culturing or PCR in centralized labs, are time-consuming and often require complex sample preparation. In contrast, microfluidic platforms can integrate sample preparation, nucleic acid amplification, and detection all on a single chip. For example, a microfluidic device might take a tiny sample from an infected leaf, perform rapid DNA extraction, amplify the pathogen-specific genetic markers using on-chip PCR, and then detect the amplified product using fluorescent markers, all within a matter of minutes. One concrete example involved a portable microfluidic diagnostic tool developed for detecting fungal pathogens in wheat. This device was able to process a small leaf sample, detect the presence of fungal DNA, and deliver results on-site within 30 minutes, enabling farmers to take immediate action. Such rapid point-of-care diagnostics can drastically reduce the time between infection, detection, and intervention, thereby minimizing crop losses and reducing the reliance on broad-spectrum chemical treatments.
Some critics claim that these on-site diagnostic devices might produce false positives or negatives due to contamination or interference from the complex plant matrix. How do you address these concerns scientifically?
Contamination and interference are indeed valid concerns, and rigorous design and validation are essential to minimize these risks. To address potential contamination, microfluidic devices are designed with integrated fluidic isolation and use disposable cartridges where necessary. The materials and surfaces in contact with samples are carefully selected to prevent non-specific binding and cross-contamination. Additionally, many devices incorporate internal controls, both positive and negative, to validate the results of each test. For instance, an internal amplification control can confirm that the PCR reaction is working correctly, while a negative control can help identify any contamination issues. Extensive field trials and laboratory validations are conducted to assess the sensitivity, specificity, and overall accuracy of these devices under realistic conditions. While no diagnostic system is perfect, the combination of robust designs, stringent quality controls, and comprehensive testing helps ensure that the risk of false results is minimized, making these tools reliable for on-site pathogen detection.
Let's talk about the interplay between microfluidics and climate smart agriculture. How can these technologies help farmers adapt to climate variability and mitigate the impacts of extreme weather events?
Microfluidics and related microscale technologies can play a crucial role in climate smart agriculture by providing precise monitoring and adaptive management capabilities. For example, microfluidic sensors embedded in the soil can continuously monitor moisture levels, nutrient content, and even temperature fluctuations. This real time data allows for adaptive irrigation strategies that optimize water use during drought conditions and prevent overwatering during heavy rains. Moreover, high-throughput microdroplet screening can be used to rapidly evaluate the performance of different crop varieties under simulated stress conditions, such as heat or water scarcity, enabling the development of climate-resilient cultivars. In regions prone to extreme weather, these technologies can facilitate early warning systems that detect rapid changes in environmental conditions, allowing farmers to adjust their management practices proactively. The integration of these systems with AI-driven decision support tools further enhances their utility, providing actionable insights that help mitigate the adverse effects of climate variability and protect crop yields.
Your explanation on climate adaptation is intriguing, but how do you ensure that these sophisticated systems remain affordable and scalable for farmers in developing regions who might benefit the most from such technologies?
Affordability and scalability are critical considerations. One approach is to design modular systems that can be deployed incrementally rather than requiring a complete overhaul of existing practices. For example, a basic microfluidic sensor for soil moisture monitoring can be manufactured at low cost using mass production techniques like injection molding. As farmers see the benefits and savings from these initial investments, additional modules such as nutrient sensors or diagnostic devices can be added over time. Furthermore, partnerships with government agencies, non-governmental organizations, and international development programs can help subsidize the adoption of these technologies in resource-limited settings. Open-source designs and community-driven innovation also play an important role, as they allow local adaptation and cost reduction through collaborative development. Ultimately, by focusing on cost-effective, scalable solutions and providing robust training and support, it is possible to extend the benefits of microscale agricultural technologies to even the most resource-constrained environments.
I want to probe further into the integration of microfluidic data with larger farm management systems. How do you envision the data from microscale sensors being integrated into a holistic farm management system? And what kind of decision-making algorithms are being developed to process this data?
The integration of micro-scale data into holistic farm management systems is a major focus of current research. The idea is to create a network of sensors, each capturing specific environmental parameters such as soil moisture, nutrient levels, temperature, and pathogen presence that continuously feed data into a centralized platform. This platform uses cloud computing and edge processing to aggregate and analyze the data in real time. Advanced decision-making algorithms, often based on machine learning and predictive analytics, then interpret the data to provide actionable recommendations. For example, if the system detects that a particular section of the field is experiencing moisture stress, it can automatically trigger localized irrigation adjustments. Similarly, if nutrient levels fall below optimal thresholds, the system might recommend a targeted fertilizer application. These algorithms are trained on historical data, weather forecasts, and crop performance models to optimize decision-making. The goal is to create an integrated system that not only monitors conditions continuously, but also learns from the data to make increasingly accurate and efficient management decisions over time.
I must ask, though, what safeguards are in place to ensure that the recommendations from these algorithms are robust? There's a risk of over-reliance on automated systems that might not account for all the nuances of field conditions.
That is an important consideration. Robustness is achieved through several layers of safeguards. First, the algorithms are developed and validated using extensive data sets that capture a wide range of field conditions and crop responses. This helps ensure that the models are not overly sensitive to outliers or anomalies. Second, many systems incorporate a human-in-the-loop approach, where the algorithm's recommendations are reviewed by experienced agronomists before being implemented. Third, redundancy is built into the sensor network so that decisions are based on data from multiple sources rather than a single sensor, reducing the risk of errors due to faulty readings. Lastly, continuous monitoring and feedback loops allow the system to self-correct if discrepancies between predicted and actual outcomes are detected. These measures help ensure that while automation plays a significant role, expert judgment and robust validation processes remain central to the decision-making framework.
Let's move on to a more futuristic topic. What emerging research do you find most exciting in the field of microfluidics and its application to agriculture? And how might these breakthroughs further transform the industry in the coming years?
One of the most exciting areas of emerging research is the development of multifunctional microcapsules that combine controlled release with embedded sensing capabilities. Imagine a microcapsule that not only delivers a nutrient or pesticide, but also monitors the local environmental conditions and reports back on its release profile and effectiveness. This kind of smart capsule could provide real-time feedback on how well a treatment is working and even trigger additional responses if needed. Another promising area is the integration of microfluidic systems with genomic technologies. By combining high-throughput microdroplet screening with next-generation sequencing, researchers can rapidly correlate phenotypic responses with genetic data, accelerating the breeding of stress-tolerant or high-yield crop varieties. Additionally, the development of fully autonomous field-deployable microfluidic labs, small, rugged devices that perform on-site diagnostics and environmental monitoring, is another breakthrough that could revolutionize precision agriculture. These advancements, along with ongoing improvements in material science and IoT integration, promise to make agricultural practices more efficient, resilient, and sustainable in the near future.
Some of these innovations sound almost too futuristic to be practical. What do you say to those who argue that such high-tech solutions are more suited to academic labs than to real-world farming?
It's a fair question, and the skepticism is understandable. However, the history of technology in agriculture shows that many innovations initially developed in the lab eventually become indispensable in the field. For example, GPS-guided tractors and precision irrigation systems were once considered experimental, yet they are now widely adopted. The key is to gradually adapt and scale these technologies in ways that meet the practical needs of farmers. By starting with targeted pilot projects demonstrating clear benefits such as yield improvements, cost reductions, and environmental gains, and then iterating on the design to improve robustness and ease of use, these high-tech solutions can transition from the lab to the field. The goal is not to replace traditional practices entirely, but to enhance them with data-driven insights and precision tools that make farming more efficient and sustainable over time.
I'd like to challenge you on a more philosophical note. Agriculture has long been rooted in tradition and experience passed down through generations. How do you balance the integration of cutting-edge micro-scale technologies with the wisdom of conventional practices that have been honed over centuries?
That's a profound question. The integration of modern technology with traditional agricultural wisdom is not about replacing one with the other, it's about complementing and enhancing existing practices. Farmers have an intimate understanding of their land, crops, and local conditions that is invaluable. What these advanced technologies offer is a way to quantify and systematize that traditional knowledge, turning subjective observations into objective data. For example, a farmer might know intuitively which parts of a field tend to be drier or more fertile. A network of microfluidic sensors can provide precise measurements of soil moisture and nutrient levels in those areas, enabling more targeted interventions. In this way, technology becomes a tool that augments the farmer's expertise rather than negating it. The challenge lies in designing systems that are both sophisticated and user-friendly so that the insights they provide can be easily integrated with the practical, on-the-ground experience that has sustained agriculture for generations.
Let's circle back to the issue of reliability under extreme conditions. With climate change leading to more unpredictable weather patterns, how do these microscale systems cope with extreme fluctuations, and what research is being done to ensure their resilience?
Resilience under extreme conditions is indeed a top priority. Microfluidic devices and microcapsule formulations are being actively researched for their performance under a wide range of environmental stresses. For instance, In the development of microcapsules, researchers are exploring materials that are not only robust under normal conditions, but also resistant to rapid temperature shifts, high humidity, and UV exposure. Some studies have focused on hybrid materials that combine synthetic polymers with natural components, providing both durability and environmental responsiveness. On the sensor side, microfluidic devices are being designed with built-in redundancies and adaptive calibration systems that allow them to maintain accuracy even when conditions deviate significantly from the norm. Field trials in regions with extreme weather patterns are an integral part of this research, ensuring that the devices can withstand the rigors of real-world deployment. By continuously refining these systems based on empirical data, we're working towards solutions that not only enhance agricultural productivity, but also remain reliable in the face of climate-induced challenges.
I'm interested in the quantitative aspects of this research. Could you share any specific data or statistics from recent studies that illustrate the performance improvements achieved by these microscale technologies compared to conventional methods?
Certainly, let me give you a few examples from recent studies. In one trial involving microencapsulated nitrogen fertilizers, researchers observed a yield improvement of approximately 15% in cereal crops compared to conventional fertilizer applications. Additionally, nitrogen runoff was reduced by nearly 30%, which has significant implications for environmental protection and regulatory compliance. In another study focusing on microdroplet-based screening for drought tolerance, the use of controlled microenvironments enabled the rapid identification of seed varieties with up to 20% better germination rates under water-limited conditions. These quantitative improvements are not isolated. Multiple studies across various crop systems have demonstrated similar benefits in terms of both yield and resource efficiency. Such data provide compelling evidence that integrating microscale technologies into agricultural practices can lead to measurable and meaningful enhancements in performance and sustainability.
Some might argue that these performance improvements, while statistically significant, might not translate into a meaningful economic impact at scale. What are your thoughts on the scalability and broader economic implications of adopting these technologies across different types of farming operations?
Scalability is a crucial factor, and the economic impact will ultimately depend on how well these technologies can be integrated into existing agricultural systems. The studies I mentioned indicate not only improved yields and reduced input costs, but also significant environmental benefits that can translate into long-term savings. For large-scale operations, even a 10-15% yield improvement can result in substantial increases in overall productivity and profitability. Moreover, the reduced frequency of chemical applications and more efficient use of fertilizers can lower operating costs. When you factor in potential savings from improved environmental compliance and reduced soil degradation, the economic benefits become even more pronounced. Of course, the initial investment might be higher, but as production scales and costs decrease through economies of scale, the return on investment becomes very attractive. Additionally, governmental incentives for sustainable agriculture and technology-driven practices can further enhance the economic feasibility, making these advanced solutions accessible to a wide range of farming operations, from large industrial farms to smaller family-run enterprises.
I'd like to explore the topic of interoperability. In many agricultural settings, multiple technologies are deployed simultaneously. How do microfluidic devices and microcapsule systems interface with other modern technologies such as satellite imaging, drone-based monitoring, and traditional farm machinery?
Interoperability is essential for creating a truly integrated agricultural management system. Microfluidic devices and microcapsule systems are increasingly being designed with standardized communication protocols that allow them to interface seamlessly with other technologies. For instance, data collected by on-field microfluidic sensors can be integrated with satellite imaging and drone-based monitoring systems to provide a comprehensive picture of field conditions. Advanced software platforms can overlay sensor data with high-resolution imagery to identify localized issues such as nutrient deficiencies or pest infestations. This integrated approach enables farmers to make informed decisions that take into account both microscale measurements and macroscale observations. Furthermore, many modern farm machinery is already equipped with GPS and IoT connectivity, which facilitates the implementation of variable rate applications based on data from microscale sensors. In short, the goal is to create a unified ecosystem where data flows freely between different components, allowing for precision interventions that optimize every aspect of crop management.
I heard arguments that these systems might become obsolete as new technologies emerge. How do you respond to concerns about technological obsolescence, particularly given the rapid pace of innovation in fields like AI and IoT?
Technological evolution is indeed rapid, and it's important to design systems that are flexible and adaptable. One of the key strategies is modularity, developing microfluidic and microcapsule systems that can be upgraded or modified as new technologies emerge. For example, the sensor modules in a microfluidic device can be designed to be replaceable, allowing for the integration of the latest advancements without needing to replace the entire system. Additionally, using open standards for data communication ensures that devices remain compatible with newer platforms and software. The field is moving toward a model where continuous improvement is the norm rather than a one-time installation. Far from becoming obsolete, these systems are designed to evolve incrementally, incorporating the latest research and technological advancements. This approach not only extends the useful life of the equipment, but also ensures that farmers always have access to the most advanced tools available for sustainable agriculture.
Let's address one more challenging question. There's been some concern about the potential environmental impact of the materials used to fabricate microfluidic devices. How do you ensure that the production and disposal of these devices are environmentally sustainable?
Environmental sustainability is a paramount consideration in the development of these technologies. Many microfluidic devices are fabricated using materials like PDMS, which, while widely used in research, are being reevaluated in terms of long-term environmental impact. Researchers are actively exploring alternative materials that are both high performance and environmentally friendly. Biodegradable polymers, recycled materials, and greener manufacturing processes are all areas of active research. In addition, efforts are underway to design devices that are modular and reusable, thereby reducing waste. Life cycle assessments are routinely performed to evaluate the environmental footprint of these devices from production to disposal. The goal is to create systems that not only enhance agricultural productivity and sustainability in use, but also minimize their own environmental impact over their entire life cycle. This holistic approach ensures that the benefits of microscale technologies are not offset by negative environmental consequences related to their production or disposal.
Before we conclude, I'd like to hear your final thoughts on how microfluidics, microdroplets, and microcapsules will shape the future of agriculture over the next decade. What are you most excited about, and what do you see as the most significant challenges moving forward?
I'm genuinely excited about the convergence of these microscale technologies with digital agriculture. The prospect of having real-time, high-dissolution data from microfluidic sensors across an entire farm, combined with AI-driven decision-making, represents a paradigm shift in how we manage crops and natural resources. The ability to precisely tailor nutrient and pesticide applications to the actual needs of the plants, while also rapidly screening and breeding for stress resilience, holds immense promise for increasing food security and sustainability. However, significant challenges remain, particularly in terms of scaling these technologies for widespread adoption, ensuring their robustness in diverse environmental conditions, and bridging the gap between high-tech research and everyday farming practices. Overcoming these challenges will require continued interdisciplinary collaboration, robust field validation, and a commitment to designing systems that are both cutting-edge and accessible. Overall, I believe that these technologies will play a pivotal role in the transformation of agriculture, making it more efficient, resilient, and environmentally sustainable.
Thank you for that inspiring and comprehensive overview. To our listeners, I want to emphasize that while the scientific details can be complex, the bottom line is that these innovations have the potential to make a real difference in how we produce food. Whether it's through improving yields, reducing waste, or protecting our environment, the integration of microfluidics, microdroplets, and microcapsules into agriculture represents a significant step forward. I have one final question. What advice would you give to young researchers or entrepreneurs who are interested in pursuing careers in this field? What are the key areas where you see opportunities for innovation and impact?
For young researchers and entrepreneurs, I would say that this field offers tremendous opportunities to make a difference at the intersection of technology and sustainability. My advice is to embrace interdisciplinary collaboration. Don't limit yourself to a single field, be it engineering, biology, chemistry, or data science. The most groundbreaking innovations are often born from the synergy of different disciplines. Focus on solving real-world problems, for instance. Work on projects that directly address the challenges faced by farmers, such as water scarcity, soil degradation, or pest resistance. Stay curious and be persistent in your research, as many of these technologies are still in their early stages of development. Also, be open to continuous learning and adaptation as the landscape is evolving rapidly. Finally, always consider the broader impact of your work. Not only the technical performance, but also the economic and environmental sustainability. The future of agriculture depends on solutions that are not only innovative, but also practical and accessible to those who need them most.
I appreciate that perspective. It certainly provides a roadmap for those looking to make a meaningful impact. As we wrap up, I'd like to thank you for sharing your deep insights and expertise with us today. Your discussion has provided a clear view of the scientific principles, practical challenges, and future opportunities in the world of microscale agricultural technologies.
Thank you for having me. It's been a pleasure discussing these topics in depth, and I'm excited about the future of this field. I hope our conversation today inspires further innovation and collaboration among researchers, industry professionals, and farmers alike.
And to all our listeners, thank you for tuning in to this in-depth exploration of microfluidics, microdroplets, and microcapsules in agriculture. Remember, the smallest innovations can have the biggest impact on our world. Stay curious, stay innovative, and keep pushing the boundaries of what's possible in agriculture. Until next time, happy farming and continued scientific exploration.
This concludes our discussion for today. We've covered everything from the fundamental scientific principles underlying microfluidic devices to the practical challenges of deploying these systems in the field, and even looked at real-world examples where these technologies have already made a significant impact. From controlled sea-label fertilizers that boost crop yields and reduce environmental runoff, to high-throughput microdroplet screening systems that accelerate the breeding of stress-resilient crops, the potential applications are vast and transformative.
As we look ahead, the integration of these microscale technologies with digital platforms and IoT networks promises to usher in an era of truly precision agriculture. Imagine a future where every plant's health is monitored in real time by a network of sensors, where data analytics guide every decision from irrigation to fertilization, and where the genetic potential of crops is fully realized through rapid, high-throughput screening. This is not science fiction. It's the trajectory of modern agricultural innovation. The interdisciplinary collaborations among engineers, chemists, biologists, and data scientists are already paving the way for these breakthroughs. The challenges are significant, but the rewards, in terms of increased food security, environmental sustainability, and economic viability, are even greater.
It's important to remember that the transition to these advanced systems won't happen overnight. It will be a gradual process that builds on both traditional knowledge and cutting-edge technology. As researchers continue to refine microfluidic devices, improve the robustness of microcapsule formulations, and integrate these systems with advanced data analytics, we will see more and more real-world applications that deliver measurable benefits. Pilot projects, field trials, and collaborations with farmers are crucial in this journey. They provide the necessary feedback loop that helps refine the technology, making it more user-friendly, resilient, and effective in diverse agricultural settings.
One of the most exciting prospects is the potential for these technologies to help mitigate the effects of climate change on agriculture. With more unpredictable weather patterns and increased incidences of drought, heat, and flooding, the ability to precisely monitor and adjust to environmental conditions will be critical. Microfluidic sensors can provide real-time data on soil and plant health, while microcapsules can ensure that nutrients and protective agents are delivered at the optimal time. This synergy not only improves crop resilience, but also helps conserve resources, making agriculture more sustainable in the long term.
For those in the research community, the future is rich with opportunities. There is a pressing need for innovative solutions that address the multifaceted challenges of modern agriculture. Whether it's developing new materials for microcapsules that are both environmentally friendly and highly effective, designing microfluidic devices that can operate reliably in harsh field conditions, or creating advanced algorithms that can process and interpret the vast amounts of data generated by these systems, there is a role for every scientist and engineer in this exciting field.
Moreover, the economic incentives for adopting these technologies are becoming increasingly clear. Reduced input costs, improved yields, and environmental benefits translate into tangible savings for farmers. As the technology matures and becomes more accessible, we expect to see a significant shift toward precision agriculture practices that leverage the full potential of microscale innovations. This not only benefits individual farmers, but also contributes to broader societal goals, such as food security and environmental conservation.
For entrepreneurs and innovators, this is a call to action. The market is ripe for solutions that bridge the gap between cutting-edge research and practical on-the-ground applications. By developing scalable, user-friendly systems and partnering with agricultural stakeholders, you can be part of a transformative movement that redefines how we grow our food. The challenges are real, but so are the opportunities. The future of agriculture will be shaped by those who are willing to embrace innovation, invest in research, and work collaboratively to turn scientific breakthroughs into everyday realities.
In closing, I want to reiterate the transformative potential of microfluidics, microdroplets, and microcapsules in agriculture. These technologies offer a new paradigm for managing crops, one that is based on precision, efficiency, and sustainability. They empower us to take a data-driven approach to farming, where every decision is informed by real-time insights and every intervention is optimized for maximum benefit. As we continue to refine these tools and integrate them into broader agricultural systems, we are poised to make significant strides toward a future where agriculture is not only more productive, but also more resilient and environmentally responsible.
Thank you once again for joining us today. I hope our discussion has shed light on the complex yet fascinating world of microscale technologies and their far-reaching implications for agriculture. Your curiosity and commitment to innovation are what drive progress in this field, and I encourage you to keep exploring, questioning, and pushing the boundaries of what's possible.
This has been an in-depth conversation covering scientific principles, practical challenges, interdisciplinary collaboration, and future directions. I trust that you now have a deeper understanding of how microfluidics, microdroplets, and microcapsules are not just laboratory curiosities, but are emerging as pivotal tools in the quest for sustainable precision agriculture. The journey from the lab to the field is ongoing, and every new breakthrough brings us one step closer to a more efficient, resilient, and sustainable food production system.
To all the researchers, entrepreneurs, and agricultural practitioners out there, I encourage you to keep experimenting, innovating, and collaborating. The future of farming depends on our collective ability to integrate traditional knowledge with the latest technological advances. As we continue to harness the power of microscale technologies, we are building a foundation for a smarter, more sustainable agricultural system that can meet the challenges of tomorrow.
Thank you for listening, and remember, sometimes the smallest innovations can have the biggest impact on our world. Stay curious, stay innovative, and let's work together to cultivate a better future for agriculture and our planet.